In this lesson, we will learn how to design the analysis and visualization for your authentic science learning experience. Now that you have completed your experiment and have collected your data, and you may even have started to discuss the outcomes a little, it's time to take a hard look at your data and put your hypothesis to a rigorous test. These activities are core to the scientific process and address many of the NGSS scientific and engineering practices. Before starting your analysis, you need to have a pretty good idea of the approach that you are going to take. Your approach should be based on the objectives of the experiment. What was your initial question? What was your hypothesis? And the insights you need to gain from your data to be able to test your hypothesis and provide an answer to your question. For example, if you're testing the type of food birds prefer, you'll want to know the number of birds that were present at the feeder with different seats. Or if you're curious why puddles disappear faster in the summer than in the winter, you will want to know how much water is left after a specific amount of time at different temperatures. Based on your objectives and requirements, you should develop a preliminary plan to get your raw experimental data into a form that you can analyze and draw conclusions from. This plan should guide your actions, but be flexible. You may have to change things up if the results come out different than you expected. Once you have figured out your approach, make sure that your raw data is going to be useful. You may need to clean it up and organize it first, so that you can use it easily, effectively, and correctly. This is one of those overlooked, unglamorous activities, a bit like organizing your clothes closet. But if you don't do it, your data will likely end up unused in a notebook or a computer hard drive. To help with this, add tags or useful information to your data. For example, if we ask our students to take pictures of animals and clothing items for 2 different projects, they could simply dump their pictures from the camera or device into a shared computer file. But then it quickly becomes hard to remember where the pictures are that we need, and almost impossible to search for them. Instead, we have our students tag their pictures and all of the data they capture. So now it's easy to find the information we need. Key tags to add to data include who collected it, what the data is, and when it was collected. It's often useful to add a short note on why it was collected, and if it's relevant, where it was collected. With tags added, the data you want becomes much easier to find and to organize. And you can quickly check if your data is of sufficient quantity and quality to allow you to test your hypothesis, or if you need to clean it up or scrub it before you use it. Even with the best experimental design, errors have a way of slipping in, especially with young scientists. And you may find that you didn't collect enough data, or that your data is obviously incorrect or not in the format you need. For example, we often see that our students use the wrong temperature scale. Or you may find that data has disappeared or become duplicated, especially if you have copied it from one place to another. Some of these issues may only come to light later, when you start visualizing, analyzing, and interpreting your data. But you want to catch as many as early as possible, and you need to have a sensible and consistent approach to dealing with them. Removing an outlier or otherwise changing your data is a big decision that could significantly impact the outcome of your experiment. And there are no set rules about this. You'll have to develop your own. For example, we always make sure to convert all our air temperature data to the same scale, even if it's not the scale we originally decided on. And as a class, we discuss whether or not to remove values that appear to be far away from the others. We don't usually use statistical tools like standard deviation to arrive at a decision. Rather, we try to reach consensus and have a clear and logical rationale for our actions. And if possible, we ground truth or cross-check dubious data. For example, by researching the air temperature recorded at local weather stations. Throughout the process of making data useful, technology is very much your friend. Digital data and tools make every step much easier, faster, and reliable. If you haven't collected data digitally, it's a good idea to spend some time entering it now. Spreadsheets are great tools for this, and not just for numbers. They can be used effectively with qualitative data, written notes, and hyperlinks. And you can even use them to organize other types of digital data, like images and documents. You might also want to explore more advanced data capture software platforms like Acerns Discovery Platform, ThatTag, and Organize Data Automatically. Now that you have well-organized, clean data, it's time to see what your data can tell you about your initial hypothesis. And to do this, you'll probably want to visualize it. There are many different ways to visualize data, depending on what kind of data you have and what you want to do with it. You might want to map it, create a gallery of images, but most of the time, you'll probably want to graph it. The first decision you need to make is what type of graph to use. A line graph is often the best choice if you want to track how your dependent variable changes over a range of values for your independent variable, especially if changes are relatively small and smooth. For example, if you want to look at how temperature, which is the dependent variable, changes over time, which is the independent variable. Line graphs can also be useful to compare changes for more than one group. Bar graphs also let you track how your dependent variable changes over a range of values for your independent variable. They work well if the changes are larger and not always smooth. For example, if you want to look at the relationship between student birthdays, the independent variable, and the month of the year, the dependent variable. And similar to line graphs, you can use bar graphs to compare different groups. If you don't know how your dependent and independent variable are going to behave, or even if there is any relationship at all between them, you might want to use an XY plot, also called a scatter plot. For example, if you wanted to explore how UFO sightings, the dependent variable, might change depending on the outside temperature, the independent variable. If both variables increase or decrease at the same time, they might have a relationship, or a correlation. And if there is no clear pattern, they are probably not related. The last major type of graph you are likely to use is the pie chart. Pie charts are very useful when you want to look at the contribution of different variables or groups to the whole. If you click on the graphing function of your favorite spreadsheet program, you'll know that this is certainly not a comprehensive summary of all types of graphs. But unless you're trying some advanced analysis, one of the basic ones should work well. Don't hesitate to experiment with different types of visualizing data. The right way is the one that provides you with the most insights and communicates your data clearly and correctly to others. Standard spreadsheet programs like Google Sheets or Excel make it pretty easy to create a graph from your data. But if you want something with more power and options, try apps like Plotly or Raw. No matter how you generate your graph, these are the key features you want to make sure it has. Start by confirming that you are using the right graph type for the data you have and the analysis you want to do. Your graph should have a title and the axes should be labeled. Make sure that the units of measurement are included if relevant. Have your independent variable on the x-axis and your dependent variable on the y-axis. And check that the scale of your graph is appropriate. You want to make sure that all the data appears on the graph and also that you can see how it behaves. Finally, check that you plotted your data correctly and clearly. Now that you have an awesome graph, it's time to take a critical look at your data and determine what it is telling you. A good place to start is to look for trends in your data. You might be able to spot trends by eye. Or you might want to get the graphing program to fit a trend line to your data. Google Sheets even has a cool Explore function that automatically graphs your data in a few different ways and pull out some interesting facts about it. You may find that it is hard to make sense of your plotted data, especially if you have a lot of data or a lot of different groups to compare. In this case, you might want to summarize or average your data. In most of your experiments, the best way to summarize your data will be to calculate the arithmetic mean or the average. To obtain the average of a data set, you simply sum up the value for each data point and then divide by the total number of data points. Averages are powerful because they provide an easy and intuitive way to summarize data. but they can be skewed by outliers, in which case the average probably isn't a good summary of your data. For example, both these data sets give the same average, but this is only a good summary for the blue group. It hides a lot of information about the green group because the values are so different. You can get a sense of how varied or spread out your average data is by calculating the standard deviation of the data set. In our example, the standard deviation for the blue group is 5. That means that we should expect most data values to be within 5 units of the average. However, for the green group, the standard deviation is 103. That's bigger than the average value, so we clearly need another way to summarize this data. Other commonly used strategies include finding the median or mode of a data set. The median is the number in the middle of the data set. For example, when we line up the weights of our green group, we find that the median value is 40, which tells us something new and important about our data that the average did not. The mode is the most common number in the data set. In our green group, we find that the mode is 40, which also tells us something new and important about our data that the average and the median did not. For most data sets, average, median, and mode will provide an accurate and informative summary. There are some exceptions though. For example, when you are measuring speed, rate, area, or volume. To accurately summarize these types of data, you'll need to do more sophisticated calculations that are beyond the scope of this module. Now that you have carefully and thoughtfully organized, plotted, summarized, and examined your data, you might start to see relationships emerge between your independent variable and your dependent variable, driven by changes in experimental conditions. For example, you may indeed see that there are more birds at the feeder when you provide sunflower seeds versus millet seeds. And you may see that water disappears more quickly when it was hot. Keep in mind the very big and important difference between cause and effect relationships, where you can demonstrate that a change in the independent variable directly causes a change in the dependent variable, versus correlations, where the independent and dependent variables may appear to be related, but you can't be sure if they are directly acting on each other or not. Your experimental findings and the relationships that they uncover provide the basis on which to determine whether your hypothesis is true or false. Remember that disproving a hypothesis is not a bad thing. It means that something unexpected has happened and that there are new hypotheses to develop and more research to be done. Finally, document and share your findings and ideas in class through a blog or elsewhere. Not only is this an important practice, it will increase your students' confidence, engagement, and interest.